Thank you so much, Patrick. And hello, everyone. Thank you again for joining us for the May iteration of our Catalyze webinar series. My name is Marissa Eastman, and I work on behalf of the Catalyze Coordinating Center at RTI International. It is my pleasure to kick off today's webinar on the target product profile and common pitfalls of clinical trials. And our presenters for today, Dr. Mark Levy and Dr. Elizabeth Mannick, are Directors of Regulatory Affairs at RTI International with vast and diverse experience in medicinal chemistry, clinical medicine, pharm tox, and both have review experience at the FDA, just to name a few of their impressive credentials. So we're very lucky to have both Mark and Betsy here today. So I think I will pass along to Mark without further ado to begin the webinar.
Okay. Good afternoon. Thank you, Marissa. Good morning to those outside Eastern time zone. We want to go on and get started this morning or this afternoon with target product profiles and avoiding common clinical trial mistakes. Now, we have to say that any reference to persons or companies, living or dead, is entirely coincidental. So what is a target product profile and why is it important? Well, target product profiles are used in the drug industry and academia as well to develop a product based on desired characteristics. They will eventually be used in the development of the label and the indication. And the indication is your only license in the United States to advertise and seek reimbursement. And these become very important as you get past the approval stage. The target product profiles are living documents. They are frequently revised, updated, edited, kept current, and should be started prior to the preclinical phase of research and development. They also contain the go, no-go criteria for moving a product forward. The required elements within a TPP are the indication, the dosing frequency, the route of administration, efficacy, tolerability, which includes safety, and if applicable, convenience. They are also quite beneficial when pitching your product to the venture capital community. We are going to show you today two product TPPs, one of which is for asthma. This could be a real TPP. This is not a mock example for some unknown disease. So you have three columns: variable, and then your attributes of minimal and optimal. So first, in the variable, you have efficacy, receptor binding, tolerance, convenience, dosing frequency, and route of administration. Now, efficacy obviously won't be determined during the preclinical phase, but as a minimal effect, you would want the forced expiratory volume exhaled in the first second, you would want a greater than 10% improvement. That's the minimum that would get you towards an approval. An optimal improvement is greater than 25%. As for receptor binding, the minimal you would want is a single receptor. Optimally, you would want multiple receptors, but for that you would have to determine receptor selectivity. For tolerance and convenience, you would want at a minimum to be non-inferior to current standard of care. Optimally, you would be superior to standard of care. For dosing frequency, 2 or more times daily is the minimal that would work. Once daily is optimal, both for patient convenience and for selling to venture capital, where once daily dosing is the gold standard. And for the route of administration, minimally effective, you would take oral. Optimally, it would be inhaled. One more example would be for pulmonary arterial hypertension, where among your priority attributes for efficacy, you would want a greater than 10% improvement in the 6-minute walk distance. And this is a standardized measure. Optimally, you would want greater than a 25% improvement. And if you were going into the European Union, you would want to see an improved time to clinical worsening. FDA's preferred efficacy measurement is the 6-minute walk distance. EMA, in this case, would prefers improved time to clinical worsening. So you'd have to work this out with the FDA, and you might wind up with co-primary endpoints. If you just had improvement in the 6-minute walk distance, you would probably have trouble with EMA. For the population who could take the drug, at minimum, you would want it to be efficacious in adults. Optimally, it would be adults, children, and women of childbearing potential. Tolerance and safety go back to the same as for asthma, non-inferior versus, and optimally you would be superior. Dosing frequency, multiple times a day, optimal once daily. Route of administration, yes, in the most serious of cases, IV, then oral. Optimally, you would have an inhalation product. So let us switch now from showing TPPs to clinical trial design considerations. The reason for this is I want to get you all to thinking about how do you study a drug clinically? Well, there are about 3 ways to do it. One is monotherapy. The other is add on to a current standard of care. And lastly, it is a non-inferiority or superiority to standard of care. With monotherapy, you do get a true comparison with a placebo. And it is ideal for testing a new agent with efficacy similar to, but with fewer or less severe side effects than standard of care. You are allowed to include patients intolerant of the standard of care. And it's justified only in short duration evaluations. The cons are that it will not evaluate drug-drug interactions or the side effect profile with standard of care. You do run the risk of a placebo effect. Patients on current standard of care will be excluded from participation. There will probably be a delay in starting standard of care for newly diagnosed patients, which means you don't have long to study the drug in patients. And as the duration of the study increases, it will require a stronger rationale to exclude standard of care. Now, as an add-on to standard of care, you don't have the ethical concerns about the use of a standard of care. It does permit the evaluation of drug-drug interactions and side effects, and it allows you to study potential additive or synergistic efficacy effects. Add-on to standard of care is frequently used in asthma as well as oncology. The downside of an add-on to standard of care is that potential improvements in the safety and tolerability profile of the new agent may not be discernible over those of the current standard of care. You really have to coax it out. Now, non-inferiority or superiority to standard of care puts the experimental drug up against standard of care. you really will know efficacy. It's either as good as or better than. And you don't have the ethical concern of a placebo-only arm. For superiority, you have a narrow window for improvement versus standard of care. And in a non-inferiority study, the FDA controls the margins, the non-inferiority margins, and those control the size of the study, which ultimately controls the cost of the study. So all these considerations can be looked at phase 1, phase 2, and phase 3. So let us first begin with a phase 1A. Well, that's also known as the first in-human study and is the first time, or should be the first time, that the drug has been into humans. What are some issues encountered at phase 1A? Well, there are formulation issues. This also includes bioavailability across oral and IV dosage forms, or lack thereof. Hopefully not, but it has happened. You have product stability. The trial got delayed and the material, you only have stability dating for so long. So you do run the risk of exceeding the expiration date. You may have a drug with a narrow therapeutic index. The immunogenicity can start to show up. This includes cytokine storm or cytokine release syndrome. It is more common with proteins. It can be fatal. And it almost sunk a large CRO who was conducting a first-in-human study. There is the determination of which you should use, of a single ascending dose and multiple ascending dose, and if there is a food effect with an oral treatment. And you have to look at the pharmacokinetics, Cmax, Tmax, area under the curve, and the half-life. I pause here to tell a brief story of what happened with a biologic. And if you're a physician listening, you know that biologics are often dosed alongside antihistamines to reduce the risk of allergic reaction. Well, a patient in the hospital had anaphylaxis post-biologic requiring epinephrine. The vial of biologic was returned to the manufacturer. What happened? This should not have occurred. The manufacturer confirmed that the vial was authentic. The lot number matched up to those sold on or about a certain date. And because the manufacturer did exactly what they were supposed to do, they had a vial of the same lot in storage, and they pulled the vial, sent the 2 vials to the analytical chemistry lab, and performed size exclusion chromatography, and found that the vial from the hospital showed high levels of clumping in the form of trimers and aggregates, while the stored vial did not. So let's work backwards. Let's go back to the hospital. Let's ask the pharmacists and the nurses, how do you get vials of biologic from the pharmacy to the wards? Well, the answer became the hospital pneumatic system. It turns out that the G-forces on the way up and on the way down of the little holder that holds the medicine that travels through the pneumatic system, those G-forces were high enough to cause protein clumping, and the protein clumps caused the anaphylactic reaction. And from then on, biologics were hand carried by technicians from the pharmacy to the wards. So while James Bond had shaken, not stirred, in this case, gently swirled is far better than shaken. Now, let's go back to phase 1B. 1B is where you start to look at efficacy in a clinical trial. So what do you have at 1B? Well, you have drug-drug interactions that can show up. you have endpoints like pharmacodynamics, validated biomarkers, safety. In rare disease and oncology, you have just strict survival. The problem is that the animal model is not often always representative of human disease and cannot always predict immunogenicity in humans. That said, there is also the issue of an expanded access protocol, which could make an entire webinar by itself, and it's often used in the rare disease space or in a public health emergency. An expanded access protocol reduces the risk that somehow there is or could result in an illegal administration of the trial drug to patients not enrolled in the trial. This is the downside of a phase 1b trial, because you have a situation in the rare disease space where if there are no drugs and you have a potential drug, but the patient either could not get in the trial because of the enrollment criteria or for some other reason, you have patients who really want in the trial and can't get in. There can be pressure to slip the drug to the patients not enrolled in the trial, and it's illegal. And so an expanded access protocol is a way to do it legally. And for that, you talk to the FDA. In exceptional circumstances, you can skip phase 2. It takes the FDA's sanction, and in the phase 1b, the study, you have to assess it for statistical power. It probably will not be adequately powered for subgroup analysis, but you can work to skip it. You'll have to work with FDA on model-informed drug development. Yes, you will need FDA's approval, and you will need an investor to be willing to put up the money for what is ultimately a pivotal trial once you've been through phase 1b that is sufficiently powered. It will be expensive. And if you're going to venture capital, they might not be so willing to give you enough money to do a full phase 3 pivotal trial. So even if the FDA sanctions it, your bankers may not go along. So skipping phase 2, you do it at your own risk. It has its benefit in that in some cases you can cut about 2 years off of your development plan, but you have to think long and hard about doing it, and you have to do it. You have to decide to try that before phase 1b, because the 1b's got to be powered, and you've got to have a large enough 1b trial that you can go to pivotal, and you got to have your investors put up money then for the 1b. So it's really everybody sit down, think about it. Let's look at the pros, let's look at the cons, talk to your consultants, talk to your bankers, and talk to the FDA. And anybody who suggests you don't is generally the one who's going to call the shots. For the next part, which is the phase 2 clinical trial planning, my colleague, Dr. Mannick, will pick up on that slide.
So in terms of phase 2 clinical trial planning, as you design your trial, you'll have a number of considerations. First, what is your trial population going to be? You have to think about inclusion and exclusion criteria. You're going to want to show some efficacy and not terrible safety in this phase. So you're probably not going to want to enroll your very sickest patients in all cases for phase 2. But you do want to have sufficient diversity. You want to think about trial size and power, typically between 200 and 300 patients in phase 2. You want to think about location, usually maybe a dozen, 10 to 15 sites. You need to think about whether you're going to just do US trials or multinational trials. If you move abroad, you're going to need to become familiar with the regulations in the countries in which you're operating, which are not always harmonized with FDA. You also need to think about the duration. Your investors are going to want you to stay short for given expenses, but typically these are 3 to 6 month trials, sometimes with extensions, and you really need to think about preclinical tox coverage for longer trials, which you may need to be running preclinical studies concurrently. And finally, you can have a phase 2A proof of concept study, usually at one higher dose, and then a phase 2B dose ranging study. But you can also run these together as a seamless phase 2A, phase 2B trial. You need to think about your study arms. Is this placebo-controlled? Is it against standard of care? Mark discussed this. Or is it even open label at times for a phase 2A or a rare disease? And then you're going to need to think about interim analyses. Again, investors and CEOs love interim analyses because you can get an early read on efficacy, but they also spend your alpha. And so they reduce the power of the final results of your trial, and you're going to have to get significance at a lower p-value. So you need to think about those things carefully. As far as pediatric plans, Mark, can you go back? It's good to start early. In the United States, the initial pediatric study plan is due to the FDA by the end of phase 2. But in Europe, you have to get a pediatric investigation plan, or PIP, submitted by the end of phase 1. So you need to be thinking about whether or not you're going to have juvenile tox studies. A lot of sponsors put adolescents into their phase 2B trials, which is great, but you need to show a prospect of direct benefit for pediatrics by showing efficacy in adults before you undertake pediatric dosing per FDA. You need to think a lot about CMC issues, and particularly bridging. Are you going to bridge the formulation? Are you going to bridge the manufacturing site or manufacturing processes? And if you are, it's really optimal to do that before the end of phase 2. And you probably need to talk to FDA beforehand. Next. Other things, don't forget clinical outcomes assessments, which include PROs, patient-reported outcomes, and biomarkers. Have you validated them? What's your plan for validation in phase 2? And is your validation what we call fit for purpose? Even if your biomarker has been validated in another disease state, you're probably going to have to revalidate it in your disease and population. And regulatory strategy. Everyone knows about the end of phase 2 meeting, but you may need other meetings in phase 2, like type C clinical meetings, CMC meetings, and especially COA meetings. And you need to become aware of indication-specific guidances for your disease, and of course, discuss phase 3 endpoints with the FDA. Finally, the target product label. By the end of phase 2, you should start to create a target product label from the TPP. It will evolve before the final label, but it's good to get something in place. And the clinical trial diversity plan. This is now mandated by law and due by the end of phase 2 to FDA. Next. So there are many, many ways that clinical trials can go off the rails, some avoidable, and we're going to discuss a number of examples, and some unavoidable. Okay, so our first example is failure to test a drug in a validated animal disease model. So why is this important? In phase 2, it's the typical time to apply for orphan drug designation and fast track designations. Orphan drug can give you 7 years of exclusivity in addition to the exclusivity you already have, and can make a huge difference in your bottom line post-marketing. And fast track, although not quite as advantageous, is important to your investors. So usually you don't have enough clinical evidence in humans early in phase 2 to get either one of these. So animal efficacy is key. An example is that a company could not get either ODD or fast track because their preclinical efficacy studies in an animal model of human disease were conducted using a backup compound and not the compound that eventually went to clinical trials. So a big lost opportunity there, although they did plan to reapply during phase 3 for orphan drug. Next. Another is ignoring that a phase 2b trial might be needed as supportive evidence in a marketing application, and therefore should be powered and conducted like a pivotal trial. So an example is a company's drug had 1 successful and 1 failed phase 3 trial. Their phase 2b trial was successful, but underpowered and used an unvalidated pharmacodynamic biomarker as an endpoint. So the 2b trial data could not be used as supportive evidence and save the drug approval. How did this happen? The company was overconfident that they could win in phase 3, and they didn't think they would need to rely on their phase 2 results. So best to have a plan B and consider using your phase 2b trial as supportive evidence. Next. Finally, failure to set CMC specifications before or during phase 2. An example is a company had a difficult tech transfer and a very innovative botanical drug. The drug concentration varied by a factor of 3 going into phase 2 studies. The company thought it was too early to set tighter specs. The FDA disagreed and put them on CMC clinical hold. Why did this happen? The company was afraid to talk to FDA. They thought what they don't know won't hurt us. But the CMC group in particular hadn't discussed the drug classification with FDA. They assumed it was just a botanical, but it turned out that it was a modified botanical that FDA considered to be a biologic as well, with different regulatory implications. The lesson being, talk to the FDA early. Next. Failure to have design controls for a digital device prior to phase 2. Why does this happen, or why is this important? You can't validate a device or a biomarker in phase 3 if you want it to be a primary or key secondary endpoint on your label. An example is a company had a digital device that they were testing in phase 2b without design controls, and they hadn't agreed on a clinical and analytical validation plan with the FDA, ending up in the project being scrapped. How did this happen? The company wrongly thought they could conduct studies under a research use only investigational drug exemption, investigational device, exemption, and fly under the FDA radar. But they still wanted to put the results on the label. And this is like having your cake and eating it too. Many people try to use a research use only label to sort of inadvertently get their drug out on the market, but they could be liable to FDA enforcement actions, and they certainly won't be able to make any labeling claims if they do that. Next. Finally, a common mistake is failure to accurately assess the risk of a device. So an example is a company had a digital device that did not pose any physical risk to the patient, but was intended to diagnose a disease. They met with FDA and suggested that this was a class 1 or low risk device. And the FDA said, no, it's a class 3 or highest risk device. Why did this happen? The company did not consider that misdiagnosis could be a risk or a significant risk in their risk assessment. Next. Failure to oversee data quality and integrity. An example is a company had a primary histologic endpoint. Slides were read by a famous overseas pathologist who liked to brag about his high inter-reader reliability or kappa scores of the pathologists on his teams who were independently and blindly reading the slides. It turns out the expert was rereading his colleagues' scores and changing any that he didn't agree with. Why did this happen? Inadequate sponsor oversight and inadequate understanding of data integrity, and failure to understand the concept of a consensus read in histology, as well as trusting experts blindly, and not obtaining FDA input about histologic scoring. Next. So for phase 3 clinical trial planning, hopefully by phase 3, your products will all have been snatched up by larger entities who will carry them forward for you. But some clinical study design issues in phase 3 are trial population. Here you want to be more representative of the final disease population who will be taking the drug. But you don't want to, again, not achieve your efficacy or safety results. So you may want to stratify by risk category. Your trial size is going to be a lot larger, and you're going to need a minimum of 1,500 patients total exposed to study drug during your clinical development program, and 100 patients exposed for 1 year for drugs that treat common illnesses. And your trial location, again, typically phase 3 trials are multi-site and multinational. Make sure you understand the country's regulations. Also make sure that FDA will accept the data from other countries. Sometimes populations are very homogeneous overseas, and you could get dinged by FDA about lack of clinical trial diversity. Duration, most phase 3 trials are 1 year or longer. And again, same issue as in phase 2 about interim analyses. If you're going to do a number of interim analyses, just make sure that you talk with your statisticians and that you power your study for taking into account the fact that you're going to lose alpha by conducting these analyses. Next. So a couple of examples from phase 3. Ignoring the concept of clinical meaningfulness. Why? Clinical meaningfulness to patients can be difficult to grasp because it involves psychometric analyses that are scrutinized by the Clinical Outcomes Assessment Group at FDA, which is different than your clinical division, review division. Example, a company's drug achieves statistically significant efficacy results in phase 3. The FDA did not consider the results clinically meaningful based on phase 2 patient interviews, and the company received a complete response letter, which is a rejection of their marketing application. How did this happen? The company had not done enough clinical outcomes assessment work during phase 2 to determine the clinical meaningfulness of their drug. Next. Insufficient vendor oversight and ignoring 21 CFR Part 11 quality regulations. Why does this happen sometimes? Companies assume large CROs do adequate sub-vendor oversight. So an example is a company's primary phase 3 endpoint included an MRI score. The CRO contracted a radiology vendor to read the MRIs. 3 months before BLA submission, an unblinding episode was discovered that involved readings of MRIs on 35 patients, and lack of an audit trail was also discovered. So how did this happen? The company had not conducted regular vendor audits because they thought the CRO had done it. When an audit was finally conducted by both the sponsor and the CRO, the vendor failed. The company elected not to inform the FDA, thinking what they don't know can't hurt us. And then the drug failed on efficacy. But had it actually gotten to marketing application, this could have resulted in problems at the time of a BIMO inspection by the FDA during drug review. Next. So in summary, sometimes clinical trial failures are unavoidable, but many actually are avoidable, and they cost millions and even billions of R&D dollars, and they can crush good preclinical development. Common causes are not talking to the FDA early and often, not conducting animal studies in a disease model with your lead compound, not validating biomarkers, devices, and endpoints in phase 2, not bridging CMC changes in phase 2 or before phase 3, not considering the patient voice early, and that includes clinical meaningfulness, clinical trial diversity, and PROs, inadequate oversight of vendors and experts, lack of data integrity, conducting too many interim analyses, and not having a good TPP. And you can avoid these mistakes and succeed. And thank you for your attention. And Mark and I will be happy to answer any questions. And after that, if we have time, we're going to give you a little audience poll. Thank you.
Great, thank you both. So as some of you all have been doing, if you have any questions for presenters, please continue to submit those through the Q&A window. As we transition to the questions, I'm also going to open the Catalyze feedback poll for the remainder of the session. So if you wouldn't mind providing your feedback, the project team would greatly appreciate it. Our first question from Jeremy, is convenience defined as convenience for the patient, for the provider, or either or neither?
Mark, did you want to take that?
Sure. Convenience is generally thought of for the patient. It's easy to dose. If it's self-dosed, it's easy to dose. But then there can also be convenience for the medical staff in a hospital. I've had clients who were developing a therapy that required making an emulsion, passing it back and forth between 2 syringes, and no nurse wanted to stand there for 15 minutes and do that, I can assure you. So it's convenience for anyone concerned.
Okay, Jeremy had another question. How do you define optimal for metrics where greater is always better with no upper bound?
I'd say just it's optimal. It doesn't have to be, you don't have to put a definition. It's just, it's optimal. It's in a perfect world. You can set it as high as you want. In a perfect world, every disease would be treated with 1-a-day oral dosing. And during allergy seasons, antihistamines would last for a week. You know, you can set optimal at anything you want it to be. Your investors in marketing will love it if you can get there.
Okay, next question. Is preclinical tox done before clinical trials or during phase 2?
So a lot of preclinical tox is conducted before phase 1, but it doesn't just end at phase 1. It continues. So usually sponsors are thinking ahead to phase 2, where they're going to need 6 to 9 month studies, preclinical tox studies, and they're going to need the results before they hit the 6-month mark in their phase 2 clinical trial.
Okay, another question here from Jay. If you have one drug for multiple indications, phase 1 results from one indication can be used for another indication, is that correct?
Not usually, unless the indication is very, very similar. And that's the concept of fit for purpose. A lot of people make the mistake of thinking you can just translate results from one disease into another, but it's very rare that the FDA will allow that.
The one addition to that is you can, with oncology, go in at a phase 1 IND and say for solid tumors, and you don't know what kind, because you don't know what kind it's going to work on. Oncology is an exception to a lot of the rules. Now, you can use it for safety. If you've got healthy volunteers in your phase 1 and you're going to look at it for another indication, you definitely use the safety population. The efficacy population, you're going to have to discuss with FDA. The safety population you can use, provided it wasn't too many years ago, because that's starting to get too close to a synthetic control arm, which could be a whole webinar by itself.
Okay, another question here. Are there differences with very lethal diseases such as ALS where survival is 2 to 5 years?
Of course, FDA looks at the prevalence and incidence of diseases and the severity. I mean, let's take the most extreme case besides the neurodegenerative, which we all know the pathway. Betsy discussed pediatric study plans, but if you have a genetic disease that affects neonates and the infants that are born are dead within 6 months, you don't have to start in adults. You can start in neonates. And that's a very high bar, but the FDA does understand the severity of genetic diseases from birth and the rate at which they are fatal. And it's just, Betsy, you want to add anything else to that?
No, but just in general in rare diseases, the game does change. I mean, that's the terrain where you may be able to skip phase 2, where you may be able to do a single phase 3. But that said, it's not a free-for-all either. And there have been some notable sort of failures in the ALS sphere because people assumed, well, this is ALS, so I can do whatever I want and I don't have to have solid evidence.
And I'll add to that. When I was on the medical school faculty, I worked on ALS research. And I can tell you from seeing human patients with ALS, one of the absolute hardest things to do is to know that you had good results in the ALS mouse model and know that the drug, when administered to ALS patients, did not do one thing. And I will tell you that is difficult. And you go back and you look and you look and you could not figure out, and you didn't do anything wrong, and you still can't figure out why it didn't work in humans. And these are patients in phase 1 who have the disease, who know they have the disease, and who are on the decline. And they will take a drug any route of administration that is possible. And you have to look at it and go, it didn't work. And I can tell you it's difficult.
Okay, next question we have here from Chris. Can you discuss some productive scenarios where, with RTI working with discovery stage, early preclinical drug discovery companies?
Mark, I'm going to let you
Okay,
address that.
we can work with you at any stage. We will be happy to look at manufacturing, even the basic chemical synthesis, your early pharmacology, your early toxicology, and not knowing anything else. I'm just going to leave the answer at that. We can sit and advise and talk to you in a one-on-one.
Okay. What kind of preclinical study may be expected to perform during phase 2 studies?
I talked about it just briefly, but a 6 to 9 month toxicology study would be typical in 2 species for phase 2. I think it's 6-month rat and 9-month dog, but don't quote me. And that's for a drug.
Okay. Brian asks, you mentioned to talk to FDA early and often. Is there anything you'd recommend not talking to FDA about?
That's a great question. FDA has seen it all and heard it all, and they will respect you if you come forward with problems. They will not judge you. What they will judge you for is if you hide your problems and then they find out about them inadvertently or too late. But of course, trying to get a meeting with FDA is not that easy. So you can't just meet with FDA every month and talk about issues as they come up. You want to try to bundle your issues together to make your meetings with them worthwhile. Mark, did you have any thoughts?
Not without knowing the actual specific product, you might not talk about every kappa corrective action plan or every out of spec measurement that you got. You might save that and learn from it, but have them in the database for when they conduct an inspection years later. At the early R&D stage, now you tell them, you disclose it. You err on the side of disclosing. Now, asking of questions of FDA, we take that offline, but there are specific ways you ask questions.
Okay, another question. I believe Jay is following up, but Jay asked, phase 1 is mostly for safety. Is that correct?
Right, but if you can coax efficacy out of a phase 1 and the FDA will let you, you try to do it. But it takes their sanction.
Okay, another question. Can you both comment on FDA consideration of sub-studies added to phase 2 trials? Is additional preclinical data here helpful too?
It depends on the duration. But again, I've seen companies run into trouble by adding extension studies on and not having sufficient preclinical coverage. So it's really about the duration, the total duration that one patient is going to be exposed to the drug. And again, you can add on sub-studies. It happens all the time, but make sure you go and tell the FDA about it before you do that. And that's where a type C meeting in the middle of phase 2 might be handy.
Okay, another question. What would be the role of formulation / drug delivery scientist to support clinical TPP?
Your formulation controls, is it IV, is it IM, or is it oral? And so some drugs start out formulated for IV and wind up being able to be dosed subcutaneously. Some are IV, but that's saved for patients who are unconscious, and oral formulations can be developed. Formulation, you can have the best drug substance, the best API you can imagine, and if you can't formulate it, forget it. You're not going to humans, or you're going to have a lot of trouble.
Okay, another question. Does TPP also apply to medical devices?
Yeah.
Can
Oh,
you
yes.
introduce a bit more on clinical trials for medical devices?
It's a whole webinar, and we have an expert, Diane Erb, who'd be happy to give a webinar. But when you run clinical trials in devices, it's different than a drug or a biologic, and you go through CDRH at FDA rather than CBER and CDER. And you usually have, rather than a pre-IND meeting, a pre-submission meeting. And there's several categories of pathways, regulatory pathways for devices. There's a 510K pathway, which is the shortest, and then the de novo pathway, which is kind of the middle pathway, and then the pre-marketing authorization pathway, the PMA pathway, which is the longest. So you're going to want to discuss which pathway you're going to take with CDRH in your pre-submission meeting. There's some commonalities between drugs and devices, but devices are a whole world unto themselves. So we have device expertise at RTI, and we'd be happy to help anybody developing devices.
And they have their own sets of guidelines, regs, and laws.
Okay, next question here is from Victoria. Can efficacy in stroke patients be counted as safety for cancer applications? Metastatic pancreatic cancer applies to other metastatic cancers or need fit for purpose studies for example, breast cancer or lung cancer.
Mark, I'm going to let you have that.
All right, I'm going to go back and look at the question. I'm going to scroll up in the question box. Can efficacy in a stroke patient be counted as safety for cancer application? It's going to depend on the situation with the patient and the clinical data, the blood work, the severity of the illness. This is one of those we'd have to sit down and look at the specific data, the specific enrollment criteria, and then talk to the FDA physicians about it. Could you possibly, with metastatic pancreatic cancer, apply to other metastatic cancers? My first reaction is probably, but you would have to make sure that what therapies the patient was on for pancreatic cancer didn't cause a drug-drug interaction with the drug you want to study. So I'm going to say probably it depends, and the oncologists and the FDA oncologists need to all sit down and have a discussion.
Okay, has NIH contracted RTI for drug development?
Or maybe I can take that one.
Cool,
So,
Mike.
That's
Thank
Mike
you.
Peck.
as part of any Catalyze award, you have access to the regulatory experts, both on this call and more specifically if you have device and diagnostics. And then we also have an opening, or within the preclinical services, there's also a standalone request and support for just regulatory support alone. So please reach out to the team if you have more questions on that. catalyzeinfo@rti.org. Thanks.
Okay, and I see one more question in the Q&A window. If there are any, we have about 5 minutes before the top of the hour, so if you have any additional questions, please continue to submit those. But what is the format for FDA conferences? I thought pre-IND meetings only happen once.
Okay. FDA conferences can occur via written responses only. They can be a hybrid Zoom meeting, video conference, and if you are in person, face-to-face. Yes, generally speaking, a pre-IND meeting is only once. However, there are type Cs and type D meetings that can be done as needed during the development process if you just have a brief question or two of FDA.
Okay, I am not seeing any additional comments, so I want to thank our two presenters for the presentation and that Q&A session. Also, I wanted to give them a chance if they had any kind of closing comments before we close out the webinar.
Well, just that thank you for attending, and we've only scratched the surface of regulatory affairs. So if you have any further questions or specific questions about your drug development program, please feel free to reach out to us.
We're happy to help anyone who comes to us, and thank you for attending. And this is just a brief overview.
Great. Thank you both so much. Just a quick reminder that a copy of the slides and the recording will both be posted on the NHLBI Catalyze website. And with that, I'd like to wish everyone a good afternoon and take care. And that concludes today's webinar. Thank you.